Hello. So I'm going to record the first part of the lecture on chapter number 1 about introduction. So I did post already a second part of the lecture. I'm gonna push the first one. So microbes are tiny but mighty. So they're both very easy and very difficult to study. They reproduce rapidly, can be grown quickly in large populations. In a laboratory, we call it culture. So the way you do it, you put a little bit of bacteria in your liquid or solid medium and put it in an incubator, which provides optimum temperature to grow. Medium contains all the nutrients bacteria need to grow. As bacteria divide by boundary fission, more and more bacteria is formed. So most of the time, bacteria cannot be seen under microscope. Well, bacteria cannot be seen directly. You need a microscope to see them. So we need, unfortunately, we need tools like microscope, and also you can analyze bacteria using methods like biochemical methods, where you uh, put a certain nutrient and then they degrade it. There will be some sort of indicator in the medium that shows color change or some other property that you can deduce that bacteria they are doing something. Okay, so the nature of microbes. Microbiology is specified area of biology that deals with living things, so ordinarily too small to be seen without magnification. So microorganisms include different classes like bacteria, archaea, protozoa, fungi, helminths, algae, viruses, and prions. Out of all these guys, viruses and prions are acellular, means they do not have cellular structure. Helminths are parasites that live inside animals, humans, or plants. They are most of the time visible with the naked eye. However, some of their stages of their life cycle are invisible, like eggs and larvae. That's why it's all helminths are part of microbiology. Microbes have shaped development of Earth habitats for billions of years. So we're gonna go a little bit into history of life. So single-celled organisms appeared on the planet about 3.8 billion years ago. The planet itself is about 4.5 billion years old. Cell types arose from a single, which is not living or extinct common ancestor. We call them last universal common ancestor. So we do have, um, we divide the all living things by the way they structured into eukaryotes and prokaryotes. Eukaryotes have true nuclei. They include plants, animals, protozoa. They have the fancy cell that has a nucleus with DNA inside and has organelles like mitochondria and chloroplasts. Prokaryotes include bacteria and archaea. They're both single celled, they have no true nucleus. However, archaea are evolutionarily distinct from bacteria. So when we're talking about taxonomic division, like talk about genus, species, family, and so forth, the prokaryote eukaryote is not a taxonomic division. We have three domains: eukaryotes, archaea, and bacteria. Those are three domains of life, distinct by comparing their DNA to each other. Okay, so that's what we were just talking about. Bacteria, archaea, and eukarya are three domains of life. They've been originated from last common ancestor, and if you can see, eukarya are they have eukaryotic structure. They include plants, fungi, protozoans, animals, so forth. They do have organelles and membrane-bound nucleus. Bacteria and archaea are two distinct domains. Means when you compare archaea to bacteria, they sometimes farther apart removed than archaea from eukarya. Although they both have no nucleus, they don't have organelles, they look very similar. The most important part to remember that based on their DNA comparison, they're two vastly separate domains. So this one is a picture that depicts a short origin of life. I want to make sure you consider this table. It shows you that according to scientific theory, we had a last common ancestor in about 3.8 8 billion years, and it gave rise to bacteria, archaea, and eukarya. Uh, and After that, we have insects, reptiles, mammals, so forth, diverged from ancient eukaryotic cells. So microbes are ubiquitous and are found deep in Earth's crust, in polar ice caps and oceans, inside the bodies of plants and animals. They're also found, they're essential to life. Um, we have many theories in science. Theory in science is usually undergo years and years of testing and have not been disproved. It's usually a well-established natural phenomenon and not just a random guess. Example of theories in science include germ theory, cell theory, and theory of evolution. So germ theory, we're going to talk about it. Literally talks about how germs are causes of infectious disease, and cell theory talks about that everything is made of cells, and each cell comes from pre-existing cells. And theory of evolution states that explains the diversity of life and literally states that accumulation of small changes that occur in organisms happen as they adapt to their environment. So literally, random changes, which are called mutation, happen in organisms, and if this change uh, encodes a beneficial character that gives this organism survival advantage, that change will be propagated in offsprings of that organism, making it more adapted to the environment. So that accumulation of changes that will eventually lead to better adaptation because it's selected by environment, documented every day in all corners of the planet, and testable by science. So we're going to talk a little bit more about major metabolic processes. So metabolism is all the chemical reaction that happens within the cell and they divided into catabolism and anabolism. So catabolism are reactions that lead to breaking down of complex things into simple. Energy is being released, and organisms harvest this energy by making it into ATP. Now, anabolism is the opposite, is the reaction of synthesis, where complex things are synthesized by using simple things, and the energy needs to be inputted. So, photosynthesis is a major metabolic reaction which has two parts. Photo means light, and synthesis means making something. So, that's literally light fueled conversion of carbon dioxide to organic material. So, we literally take carbon dioxide and we build more complex molecules like carbohydrates, like glucose, out of it. So, the light is used as a source of energy. So you guys probably heard how if you take a plant and put it under the glass dome, and you take oxygen out of that little dome, and let the plant grow, and then put a little animal in there like a rat, the rat will survive because during photosynthesis the oxygen is being produced. That's just one type of photosynthesis. It's called oxygenic photosynthesis. That's what the plants around us do. Some cyanobacteria do, and a lot of little protozoans that live in the ocean. That's how they produce oxygen. They take water, split it to hydrogen and oxygen, take hydrogen to build carbohydrates like glucose because it has atoms of carbon and hydrogen in it, and they discard oxygen as a waste product. So that's a second type of photosynthesis. It's actually originated from a more ancient form of photosynthesis, which is called anoxygenic photosynthesis. So anoxygenic photosynthesis arose first in in the Earth history, occurred in bacteria, and it did not produce oxygen as a waste product. Uh, it actually had hydrogen sulfide as a source of hydrogen and broke it off, and the waste product was sulfur. So it's actually more efficient in extracting energy from sunlight. However, as ancient organisms were doing anoxygen photosynthesis, they ran out of hydrogen sulfide, and if they didn't, there was no any other sources left, they would have gone extinct. But As we mentioned about evolution, chance mutations happened, and that allowed certain organisms switch to using water as a hydrogen donor, and that led to emergence of oxygenic photosynthesis. We still have anoxygenic photosynthesizing bacteria around. They live in anoxic environments, environments with no oxygen, and you'll be surprised how much of that we have. If you go to sediments of lakes or swamps, or even certain parts of ocean where there is no oxygen, you can find an oxygenic photosynthesizing organisms there. As long as there's light and no oxygen and donor like hydrogen sulfide, you can find them. So we're going to talk more about how we just talked about how microbes produced oxygen as a result of oxygen photosynthesis. They also produce CO2 as a result of and CH3, which is methane, as a result of degrading organic matter. Because a lot of microbes are decomposers, they decompose dead things and release all these gases. Um, so As we said, bacteria are the most abundant cell organism in the oceans and the number one photosynthesizers. What does this mean? So we know that a lot of plants do oxygenic photosynthesis, make oxygen for us. However, even if in theory, theoretically, all the plants were gone, we would still have enough marine organisms to produce oxygen for people to live on. So if we're talking about most abundant cellular organism made of cells, in this case prokaryotic cells, they would be bacteria. Most abundant acellular inhabitants of oceans would be viruses. So viruses do not are not part of tree of life because we can't compare them with the rest of the organism, because the three domain of lives are based on the comparison of 16S RNA gene, which is a part of genomes of those organisms, and they encode 16S ribosomal RNA, which is a part of ribosome. Viruses do not have ribosome, that's why we can't use them, and they do not have cells, so they are not really truly alive organisms. So when we talk about bacteria, we already mentioned that some bacteria act as primary producers. They actually make organic compounds using photosynthesis. Some of them act as decomposers because they decompose dead things, but they also, some of them live in close proximity in symbiosis with plants. And also fungi live in symbiosis with plants, and both bacteria and fungi assist plants in getting their nutrients. However, one more thing that we will stress in our class is that about 2,000 species of bacteria are pathogenic. They cause diseases of animals and plants and humans. So we want to talk more about historical current uses of microbes by humans. So as we talk about microbes, the first thing that comes to mind is a process called biotechnology, where people use microbes to produce certain value-added products like bread, alcohol, and cheese and yogurt. So as people were applying bacteria and yeast to make this above mentioned products, as our understanding of genetics advanced, people realized that they can actually take that organism and modify it genetically to make it a better producer. So manipulating of genes of model organisms actually is a hallmark of genetic engineering that allowed us to manufacture things like insulin by creating recombinant organism. What is a recombinant organism, or a GMO, genetically modified organism? Is where you have a model organism like bacterium or yeast, and you insert a gene from maybe a human which encodes insulin or any other biologics, and you grow that GMO in culture. And as this modified organism grows, it will manufacture a product that is encoded by the gene you inserted into it, like insulin. Before we could make insulin using genetically modified organisms, people had to go and harvest insulin from cadavers, which dead bodies, and that was not very efficient practice. However, now people can manufacture a bunch of insulin and for the price of $30 a vial of insulin, which is uh, can be a life supporting for somebody with diabetes type 1 or 2. So using genetic engineering biotechnology literally allows people to save lives of hundreds and thousands of people. So, as we mentioned, people use genetic engineering to create genetically modified organisms where they can use them as producers of value added chemicals like insulin. So, all this is parts of recombinant DNA technology, which allows transfer of genetic material from one organism to another, and that will literally making their DNA different to allow us people to take advantage of that and make products we want to make in mass scale. Okay, so the practice of medicine allowed us to advance our treatment of wounds and lesions, perform operations, and major discovery of antibiotics actually revolutionized medicine, allowing us to prolong people's lives, save them from infection on operation table and any other infection situations. Another way of applying organisms is in industry, like mining precious metals like gold, where organisms are used to purify gold. And bioremediation is another way of using microbes in industrial settings where um, we use microbes already present in the environment or introduce them there intentionally to restore stability of the system or clean up toxic pollutants. For example, when you have a major example is when we had BP oil spill. We had in Gulf of Mexico, we introduced a surfactant, which is really a soap, to break down the oil into tiny droplets, and bacteria that was already present in the Gulf of Mexico because it had some oil seeps. They are naturally adapted to break down hydrocarbons like oil. That breaking of the oil into tiny droplets allowed increase the surface area and allowed the microbes to do better job of breaking down the oil. Another thing we mentioned is that over 2,000 different microbes, microbial species are pathogenic. That means they can harm people, animals, or plants and cause disease. 10 billion infections still occur across the world every year. Inf- so majority of the infections are coming from respiratory infection agents like viruses, like rhinoviruses, cold viruses. Bacteria are actually behind viruses. So unfortunately, infectious diseases are still if you compare United States with the world, you can see that the pink circles indicate diseases that caused by microbes, and blue circles indicate non-infectious diseases. So top causes of death in the United States, we have top five: heart disease, cancer, accidents, low respiratory diseases like COPD, and stroke. COPD usually caused by a previous lung damage caused by smoking or maybe living in some toxic zones where there is toxin chemicals that damage lungs. However, in worldwide, we see that the pink circle, which is called low respiratory disease, it's in pink. So in this case, this low respiratory disease is not caused by smoking; it's caused by um, infectious agents like pneumonia, streptococcus pneumonia, bacterium, and other flu viruses. So it's still in within the top five causes of death. We also have diarrheal diseases, number nine, and tuberculosis, number 10. The only pink circle we have in the causes deaths in the United States is influenza and pneumonia, and it's within top 10. So if we talk about influenza and pneumonia, it's not that it kills younger people. Majority of the people it kills are people are older than 65. That's why, guys, if you ever go and how you if you although unfortunately our flu vaccine is only efficient about 70% of the time, even if everybody vaccinates, we can achieve herd immunity and protect older people from getting pneumonia because a lot of the time it's fatal for them. So, so let's kind of talk about emergent diseases. So when we talk about emergent disease, are diseases that actually emerged recently in maybe within last 20 or to 50 years. and the one of the most important ones is AIDS. We still have, although we do have very efficient medications for AIDS, people actually, while taking meds, they can live their life and not even be contagious. It's still considered a very important disease. Then we have hepatitis C, which affects liver cells. We have Zika virus. Again, we're going to talk more about it. West Nile virus and tuberculosis. So tuberculosis is not really emergent disease; it's an old disease as we see, but it is can be called a re-emergent disease because if people in certain developing countries go through maybe a conflict where you have like refugee camps and they are all housed in close proximity, tuberculosis emerges and can spread in this type of circumstances. So also, apart from talking about emergent or emergent infectious disease, there were some association found between non-infectious diseases and microbes. So, for example, everybody knew about gastric ulcers, and from 16th or so century, which is literally ulcers forming within the stomach lining. However, later in about 1970s and 80s, it was discovered that everybody who had an ulcer also had a bacterium called Helicobacter pylori, and it took a brave resident who had his doctor student, medical MD student who was in his residency. He actually tested himself, and he was negative for H. pylori. He actually grew it in culture and gave himself H. pylori infection by drinking it, and was monitoring himself. Within first few months, about 3 4 months, he developed stomach ulcers. He treated himself with a course of antibiotic, and the ulcers went away, and he's tested negative for H. pylori. So he literally proved that the H. pylori was causing ulcer together with other things. And if you treat a patient for H. pylori infection with antibiotics, the ulcer will also go away. The problem that a lot of people get reinfected with H. pylori because it's in every if you have seafood and any other things like that, it's very common. So about 2 out of 3 or 1 out of 3 people in the world infected with H. pylori, but not everybody who's infected develops stomach ulcers. Unfortunately, 1 or 2% with H. pylori who did develop stomach ulcers will go on, and if untreated, will develop stomach cancer as well. So it is still a mystery why H. pylori causes stomach ulcers and cancer in some people, but not in others. Uh, researchers think that genetic factors are involved. So other things like multiple sclerosis, OCD, coronary artery disease, and obesity have been linked to chronic infections with certain microbes. And if we modulate composition of microbial communities, we can modulate presentation of those diseases as well. So, increasing number of patients with weakened defenses are subject to infections by common microbes that are not pathogenic to healthy people. So what does this mean? We have some people with lower immune system function that are more susceptible to infection with bacteria that are not infecting normal healthy people. And also we have another trend is increase in microbes that are resistant to drugs. So as we introduce a new antibiotic in the market, it takes about 2 to 4 years for new resistant strain to emerge. So we already mentioned divisions of all living things that are non-taxonomic are eukaryotes and prokaryotes. So eukaryotes includes a single domain. They have membrane organelles and each generally larger cell size. Most are multicellular. Organelles are small double-bound structures that perform special functions. They include nucleus, mitochondria, chloroplast. So some eukaryotes are microorganisms like protozoans, for example. You need microscopes to see them. The some are microscopic, they're multicellular like animals and plants that you can see with the naked eye. And finally, we have a second group, prokaryotes, and although they do they belong to two separate domains. So if you compare their DNA, they're evolutionary distinct, and they're bacteria and archaea. They do not have organelles. The DNA is free swimming in the cytoplasm. There is no nuclear membrane around it. They're about 10 times smaller than eukaryotes. So although they do not have organelles, they have a very complex internal organization of their membrane. So membrane forms compartments which allows them to separately run processes like photosynthesis, respiration, synthesis of new parts of their cell wall. So that compartmentalization is essential, and it's found in the prokaryotic organisms. And then we did mention viruses and prions. They both are acellular organisms. So that means they do not have cellular structure. They are not made of cells. So viruses are obligate parasites. They need hosts to reproduce. They're not independently living organisms. They exist at the level of complexity somewhere between large molecules and cells. So literally, if you look at the virus, you'll see a RNA or DNA molecule that is wrapped with special structure called capsid, and capsid is made of repeated capsamere. It's almost like chain link on a night where you have this tiny uh, links connected to each other to make the chain link. So the same thing here happens in viruses. You have the chain link structure made of individual protein subunits surrounding that DNA creating protective sheath of some sort. Okay, so viruses can either have DNA or RNA. They never have both. Okay, so now prions are even weirder. Prions do not have any DNA or RNA components. They actually are a single molecule of a protein that is infectious. So what does this mean? So if that protein prion comes in contact with a normal protein from brain cells, it actually initiates the normal protein changed into this infectious protein conformation, and that conformation disrupts function of normal brain cells that actually activates certain cells inside the brain that act as immune cells, and they will consume this that deformed proteins by, and that will make holes in the brain, causing spongiform encephalitis. So we know it as mad cow disease, or also another part of this disease, another manifestation happens in humans, and it's called Creutzfeldt-Jakob disease. So again, we just went through viruses and prions. They are both acellular. They do not have cells. Viruses are made of nucleic acid plus protein, nucleic acid being either RNA or DNA, and prions are made of proteins. So as we mentioned, there are 6 types of microorganisms, including helminths, fungus, protozoans, bacterial, viruses, and prions. And although helminths can be visible for the naked eyes, their eggs are not visible. So we're going to talk about how microbes and their role in disease and other processes like fermentation were done during history. So there were 2 groups of people. They believed in so-called abiogenesis versus biogenesis. So first group believed in abiogenesis, which embraced that if you throw certain things together that are non-living, the living thing spontaneously arises. So example, they said if you open a little cupboard and you put a bunch of old clothes in there and some oil and some dirty laundry and close it, the mice will spontaneously arise from that, which is sounds like a very silly assumption, but people did truly believe it's really happened. So that was believed to be happening because there was a vital force that was just present everywhere and that was able to summon a living thing out of nowhere. So that theory was called abiogenesis or spontaneous generation. um, so that was first kind of camp, and the second camp, the other scientists said, "Well, this is BS. You can't just do that. There is no things coming out spontaneously. They usually get into whatever you put it, like a vessel or enclosure. They kind of get in there from the outside. So nothing forms from non-living things spontaneously." So there was this scientist that we're going to look at his contributions. His name was Louis Pasteur. He lived in France. He studied the roles of microbes in fermentation of beer and wine. He was actually a chemist by training, not microbiologist. And in his times, fermentation believed to be totally non-living process, like just purely chemical. He didn't know about enzymes, but he thought they just present in beer and wine. It just happened to be there and do the thing. Okay, so they thought it was like plant related or something. However, he was interested in this biogenesis versus abiogenesis, like the normal, what we now accept biogenesis. So the living things only come from another living thing. And he kind of wanted to once and for all disprove that spontaneous generation happens, and he designed special swan neck flask experiment. So he filled flask with broth and shaped the openings into long swan neck tubes. He heated the flask to sterilize the broth. So, as we said, microorganisms were already discovered way before Louis Pasteur in 1600s. So people knew there were microorganisms, and they knew if you heat the liquid, you'll kill them. Okay, so that was not the news to anybody. However, so his experiment was a novel thing, right? So after he heated the flask and all the broth was still in both flasks, he treated them differently. One flask was exposed to dust from the air, and another flask was exposed to air but not to dust, and that showed no microbial growth. So let's just kind of look at the picture and make sense of it. So this is your flask, right? He kind of like heated them, and he exposed to dust and air by breaking off this flask, and in some scenario he actually laid the flask on its side, and airborne microbes entered and growth occurred in the sterile environment in the medium. However, in another flask that was left intact, the air was still going in, right? And however, the organisms couldn't penetrate it due to the nature of this swan neck, and so the broth remained intact. So that was the last nail in the coffin for spontaneous generation. So we're going to talk a little bit more about Robert Hooke and Anthony van Leeuwenhoek. So Robert Hooke was the first scientist who studied household object plants and trees using first microscope. He described first microbes and described cell structures. So he actually named cells. So he actually observed dead plant tissue cork. That's the one that they use cork for oak trees to make corks for their wine bottles. And he happened to observe it, and he found that they structured like little cells, gel cells on top of each other, and that's why he called all cells we see cell. Okay. And guys, remember, plant cells have durable cellulose made cell walls. That's why that organization really inspired him to name them cell. But if you would have looked in animal cells, they have no cell walls, so they have a regular shape. So he might have been called them something else, right? Just kind of a serendipitous thing. And finally, so Robert Hooke was also making discoveries in physics and other sciences. He was kind of a Victorian gentleman. And we also have another scientist that lived. He was not really a scientist. He was a merchant. Anthony van Leeuwenhoek looked kind of. He was a contemporary of Robert Hooke. Was inspired by him. He was actually a fabric merchant. That means he like manufactured fabrics, and one of the quality control steps was to look in and count the fabric fibers using microscopes. And again, those microscopes were very primitive. You had to bring them super close to your eye to see anything, and it was kind of annoying him because he couldn't count. He had to use it every day, to do quality control, and he was really aggravated. And so he actually set out to make a better microscope so he can see better his fabric, right? And so because of that, and because of his natural curiosity, he actually started observing all kind of liquids and things around him. He observed animals in a drop of water, which he calls them animalcules because they were so tiny. He also scraped his teeth kind of thing, and because the back in the day people really didn't brush their teeth that much. Plus he also looked at um his sperm, okay, and he observed spermatozoids, and he also looked at his own diarrhea, guys. That's how curious this guy was. And he discovered a protozoan in his stool, which is called Giardia lamblia. And unfortunately, that protozoan is not carrying his name, but he was the first one to describe it. And Giardia lamblia is still around today. It causes infection of intestinal tract of all kind of wild animals, including beavers and other animals that live around the water. So, if in some areas of United States, like Kentucky, this disease caused by people drinking contaminated water with Giardia. It's called beaver fever. So this also happens in some parts of Colorado where people just drink water from untreated water from streams. It seems clear. However, it might carry cysts of Giardia, and as they drink it, it gets into the intestine. Small intestine develops into adult organism. It's actually plants itself onto the villi of the small intestine and reproduces to such an extent that it impairs absorption of fats. So as the food goes through the person with that disease, the fats are not being absorbed. This actually causes certain malnutrition. People might lose weight, and they produce this explosive foul smell and diarrhea, which is floating and is high in fats, and it's called steatorrhea. Okay, obviously, if their diet is high in fats, right? So Giardia lamblia was first discovered by Anthony van Leeuwenhoek. So we also have three other scientists: Ferdinand Cohn, Oliver Wendell Holmes, and Ignaz Semmelweis, and Joseph Lister. So Ferdinand Cohn kind of was working alongside Koch, Robert Koch, which who was one of the contributors to germ theory. Ferdinand Koch described heat-resistant endospores. Certain now we know endospores are special bacterial cells that have a really thick cell wall, and they can survive boiling for 4 hours. And they you only can destroy them using sterilizing methods like autoclaving, where you apply high temperature and high pressure. Oliver Wendell Holmes and Ignaz Semmelweis described the importance of hand washing in preventing disease in hospital settings. And I did tell you a story that Ignaz Semmelweis was a doctor who was working in a fancy hospital where they had three stories. In the first story, they had clinics. In the second, they had maternal ward, and they also had um, autopsy room. So a lot of the times, doctors would perform autopsies, trying to understand why a person had died, and then they would run to maternity ward to help pregnant ladies to give birth. And Ignaz was just taken by surprise because there's so many women that died of sepsis in this hospital while giving birth. Because across the street they had a kind of a midwife place where all they did was helping mothers to give birth, and they didn't have nearly as much maternal death there. So he was like, how is this possible? We have such an advanced hospital and we have many more mothers dying. And he actually figured out that when doctors come back from autopsies, they obviously didn't wash hands, and they literally stuck those hands inside if you had hard delivery, the mothers would get sepsis from those doctors. And he was trying to tell everybody to start washing hands and how it's important. And it took few decades for people to actually catch on. So that was Ignaz Semmelweis' story in the story of hand washing and story of aseptic technique being established in the hospital. So aseptic technique is a bunch of things doctors and some researchers do to prevent contamination of sterile area. So when doctor is gowned into the gown and into the mask, the surgeons are trying to prevent their own contaminant reaching sterile cuts into the human body, that operation they perform. Another example of aseptic technique is when you try to make a pure culture of your organism and you prepare the medium, you sterilize it, and when you put the microbe you wanted to grow in there, we call it inoculation. You trying to prevent any other contaminant from the air to get in there by using flaming, by using flame sterilization of your loop and things like that. So those are aseptic technique, and the first people who paved the way are Oliver Wendell Holmes and Ignaz Semmelweis. And Joseph Lister also was the one to use aseptic technique in surgery. So literally that's where we kind of stopped, and our next lecture was with germ theory of disease, where we talked about Louis Pasteur and Robert Koch. So Louis Pasteur invented pasteurization, which is a gradual heating of a solution like milk or beer to kill majority of the microorganism. You can't autoclave or sterilize milk simply because it will break down because it's a complex emulsion. Okay, so pasteurization is a special thing that kills majority of organisms in beer and milk and other food products. Louis Pasteur, we also know, disproved spontaneous generation using swamp neck experiment that we just talked about. He also conducted diverse studies linking human disease to infection. So before that, people did not really know what caused diseases. Like an example is that the word malaria meant bad air. So they thought if you inhale bad air from the swamps, you get malaria. But now we know that it's actually carried by mosquito and it's caused by a protozoan. So this research that was done to link human disease to infection to infectious agent done by Louis Pasteur and Robert Koch contributed to development of so-called germ theory that said that germs or infectious agent cause diseases in humans. So Louis Pasteur actually developed rabies vaccine. He didn't know anything about viruses. He did not know um, um, about rabies that much, but they knew that the rabies are transmitted to humans through bites of rabid animals. So Louis Pasteur did develop rabies vaccine by passing the rabies virus through different animals. So he literally took a CSF sample, cerebral spinal fluid sample from um, infected people, and he passed them through animals, giving them to animals and making virus less deadly. And then he took CSF from some of those culture animals, and they contained weakened virus, and the virus actually gave resistance to rabies and saved the life of a little boy that his mother brought it to him to Pasteur and asked for help because everybody knew he's gonna die because everybody knew rabies is incurable. But that vaccination saved his life. However, let's just make sure we don't make mistake. The Louis Pasteur was not the first inventor of vaccines. The first inventor of vaccine was Edward Jenner, but Louis Pasteur did go into his footsteps and develop rabies vaccine. And Robert Koch contributed also was important researcher, contributed to germ theory as well. And he actually established four postulates that we listed here. We have to make sure we remember them, okay? So as organelle state, the four criteria are, so first, the microorganism, the pathogen, must be found disease but not healthy individuals. The microorganism must be cultured from diseased individuals. Like you have to actually grow it in pure culture using aseptic techniques and a solid medium. Then you have to take the culture, inoculate a healthy individual, maybe through inhaling or maybe injected into blood, and that a healthy individual, we mean that maybe a healthy, maybe like a healthy lab rat or something, and the markers must recapitulate the disease, means repeat all the signs and symptoms, and if the markers must be re-isolated from that diseased animal and matched to original microorganisms. So Robert Koch showed that anthrax was caused by Bacillus anthracis, and he also was showing that Mycobacterium tuberculosum caused tuberculosis. So we're gonna stop this right here because the next lecture is gonna pick up from discoveries of restriction enzymes and things like that. So I'm gonna finish this, and we have one more little tail of this lecture that I will probably go over the next class,